So what I'd like to talk about today is just what more understanding we've got about ME/CFS in the last year, and also to talk about its relationship with long COVID, because one of the things that I think that we all hope is that ME/CFS and patients will get a lot of benefit from whatever is happening with a huge interest in long COVID. And so it is really important to know that long COVID is going to be a very closely related disease. So I'll actually speak to that. I thought, because I've now completed 10 years of intense research on ME/CFS, I'd start off with an acknowledgement slide, because that tends to get sort of put at the back and almost forgotten. And on my left, I've got people that have inspired me both here in New Zealand and also overseas. And I'd just like to really point out, I guess, two from the collaborator list. From the time that I started, Roz has just been a tower of strength to me, and I'm really grateful for our constant interactions. You know, virtually a week doesn't go by where we don't have one or two exchanges. And more recently, of course, I'd like to acknowledge Anna Brooks, who's now on the committee. Anna is as passionate about long COVID as I have been about ME/CFS, and we've kind of merged our passions, and you know, we're sort of getting going on working together. I'd also like to acknowledge just the huge support that ANZMES has given me, not only you know, and also the financial support when it's been able, and as it's turned out, very generous private donations from individuals and families. Some of these have been very large, and they've just appeared out of the blue, and just as we were thinking we wouldn't be able to sort of go on much longer, you know, it's enabled the research to go on. So this has meant that I've been able to continue to research for the full 10 years, and I value the grants I've got from some of the charities listed there. And lastly, because all my studies have been with New Zealand ME/CFS patients and now some long COVID study participants, I'd like to express the group's gratitude for all of those that have willingly participated. Now I'm an Emeritus Professor. I can only have a limited size experimental group, but so now we're a type 4 this year. But these are incredibly valuable, loyal, and committed people, and we've had a very successful year. On the left of the screen there is Tate Peppercorn, who has actually been with me for over 20 years. She's been part of my Alzheimer's research program, but towards the end of her time did a PhD and submitted it late last year and graduated this year. And she wanted to stay on and work on ME. So she's got expertise in the evaluation of large data sets. And so she's been incredibly valuable this year. On the other side of me, Anna Blair is a member of my young university woman study group, but she's also this year been doing a master's degree in genetic counselling with the University of Technology, Sydney, and they've contracted me to do her master's research project. And so she's looked at susceptibility factors for both ME/CFS and long COVID, and I'll talk a little bit about her work through the talk. And then on the right, we have Tina Edgar, who's I employed as an 18-year-old, and you can guess how old she is now, but she's been with me for 42 years. And she's just an incredibly valuable, loyal person. She's got great skills. And it's meant, you know, we've worked really well together this year and had a high level of productivity. And I just want to mention here 3 papers we published in the last 6 months. Over the last summer, I hoped always that my research would get to a point where I could write a paper on a holistic model for ME/CFS and why it's an ongoing disease and why there are frequent relapses. And so I did this with the help of a medical student, Max Walker, whose father has ME/CFS. and that was published in the Frontiers of Neurology in late May and has been very well received. Part of that probe what molecular mechanisms might be involved in going from the dysfunctional physiology to the actual symptoms. And so Max, through his third year medical year, has worked with me, and we've published another paper in the European Association of Medicine journal and focused on the significance of oxidative stress in the pathophysiology of both long COVID and ME/CFS. And then in October, ME/CFS study, and this has two unique features. Most studies around the world are just single point studies, but this is a longitudinal study where we took blood over a full year from ME patients and controls, and we were looking at what happens in the epigenetic code of the DNA. That's the code which really determines the level of expression of all of the genes. It's really where there are light switches and dimmer switches. We want to know what goes on in the life of an ME/CFS patient. And we found quite remarkably that it's very unstable compared with healthy controls. This has not been recognized before. But we also wanted to capture a relapse recovery cycle, and we were able to do that with the two patients who were in the study. And so there's individual patient studies, which is really what I'm pressing for now for ME/CFS research, internationally, rather than just large numbers where the effects tend to get very evened out. And what we were amazingly able to see, of the 3 billion bases in the DNA genome of the patients, we was able to see individual bases that where chemical modification of them was changing during the relapse, and then it would recover when they had actually gone into recovery. So that was quite an exciting study, and again, has been very well received. Now, because I think there's huge interest on and hope that long COVID emphasis and research will benefit ME/CFS, I thought I'd just give a midterm report card on that. I was very kind of optimistic and positive at the beginning. I'm still optimistic, although there are some qualifications to what's happened, which I'll actually mention in the next slide. But let's look at the positives. ME/CFS, the name of and the disease, has been mentioned much more in media coverage nationally and internationally. I mean, this has been documented in international things, although there's been a writer which says that most of the reference to ME/CFS has been rather superficial, but at least a lot more. So the name is out there. Amazingly, I have received weekly requests from print journalists, from radio stations, for journalists for TV interviews, and they've just gone on and on about ME/CFS, people wanting to find out much more about ME/CFS. These have been largely young women journalists who often have had some contact with someone, a school friend when they were at school, or maybe a cousin or whatever, and they've just been really thirsty for knowledge about the disease. So they've been very fulfilling, long interviews, most of them. For example, this week I had one from someone who's a qualified lawyer writing for Spin-Off, and she's writing an article about the potential benefits to ME/CFS from long COVID. So we had a lovely interview earlier in the week. And then just to show the interest, last night I received a request from a Nobel Prize winner who was editing volumes of a series on methodology used to study diseases and other things, and they're planning a volume exclusively on ME/CFS, and I was asked if I would edit that. So that's a task which I'll pick up for next year. So that shows you there's so much more interest. So that's a real positive thing. In terms of the research, certainly those closely connected with ME/CFS have been energized. Some have switched to work with long COVID or have included long COVID in their research portfolios. I have to say, money hasn't really come to these ME/CFS researchers, but I think everybody feels they have a lot more mana now in terms of what they've been doing. One of the positive things is clinical trials for promising drugs in the past that could, suggested be a benefit for ME/CF patients, but were never kind of taken to clinical trials. They were never approved. There's now the possibility and talk that these could occur with long COVID. Things like low-dose naltrexone, Ampligen is being talked about, and also some antioxidants. And then the indirect benefits, of course, we hope with all of the focused long COVID studies that are going on, there'll be a spillover to ME/CFS. And really, this is going to depend how closely the long COVID is as a post-viral fatigue syndrome to the umbrella group of ME/CFS diseases. And, I'll talk about that, and I'm still very optimistic about that. And of course, because the COVID has been in such large numbers, and suddenly there are all of these post-viral fatigue syndrome people with long COVID, it's just been naturally accepted as a real disease, not psychologically based, and that's given ME/CFS, I think, a real boost in terms of the acceptance of that disease. So what are some of the qualifications? Well, there was a patient in North America who's written a rather sober analysis of what it's meant for ME/CFS patients in the US. And she talks about the lack of specialist ME/CFS clinicians. And now with all of the long COVID patients, it's very difficult for ME/CFS patients to get appointments with their clinicians. Some of the specialist clinics there, which were for ME/CFS, have put new billboards up, and now they're long COVID clinics. And so ME/CFS patients aren't necessarily included anymore. And even, the Bateman Centre, which has been a core and fantastic supporter of ME/CFS research, in the quite difficult times of acceptance, has now put limits on the time that you have had ME/CFS before to be able to be actually selected in to be studied in that. So you have to have ME/CFS for less than 4 years. So that's a kind of a slightly sobering thing, that the more patients and, the limitation in the clinicians, that could be a problem. Promise funding, of course, just before the pandemic, there was a promise that the NIH was going to increase its funding for ME/CFS by a few million, and I was really excited about that in 2019. Of course, with long COVID, there's been a promise of well over $1 billion being put into funding. But so far, in the Recover project, this has only gone to projects that involve long COVID patients, not any ME/CFS patients. So we hope, of course, anything that comes out of that will benefit, but it would be nice if ME/CFS patients could actually get into studies with that funding as well. When we look at government programs, I had hoped that ME/CFS would be a role model post-viral syndrome for long COVID and be recognised in government programs. I would say so far it's been at best lukewarm. Certainly when I found out who in the ministry, our ministry, who was going to be overseeing the development of a long COVID problem, I contacted that person and talked to them about ME/CFS and the importance of actually having that linked in. And I have to say, I haven't got any commitments, or, I don't feel there's a high degree of interest in doing that. So I hope that will change. I had another experience where I talked at a national finance conference, and they wanted me to talk on the financial impact of long COVID for the financial system. And I was in the session, the first to talk, and I started off by saying that long COVID is not a new disease. It's a classic, or contains within the patient group, a classic post-viral syndrome. The difference is that there are so many cases of COVID, that there are so many cases of this, that it's the incidence of it is so much more. And the next speaker was the head of the CEO of a Crown research entity which has been involved in the COVID thing, and he got up and challenged my view that long COVID wasn't a new disease. So there's kind of, continuing work to be done. And so, and particularly in the last point I've got there, is to make sure ME/CFS is being included together with long COVID in planned social response programs. And I'm certainly working on that at the medical school here in terms of medical education. So the important question then is, are long COVID and ME/CFS very similar diseases? And I think it was a really good start when the World Health Organization in October 21 came out with a clinical case definition for long COVID. And it was done from an iterative process with 250 people contributing, many patients as well as experts. And, for a change, there was a majority of women involved in this process, because long COVID is also more women affected than men. And they started with a constellation of about 100 long-term symptoms that were kind of recognized, and distilled out those symptoms which were present, thought to be present in most patients. So these were the core symptoms, and fortunately the clinical case definition with those core symptoms is remarkably similar to the more modern clinical case definition for ME/CFS, which are generally accepted. That is fatigue, post-exertional malaise, brain fog, unrefreshing sleep pain, and the one that comes up in long COVID at a higher incidence, of course, is breathlessness, and that relates to the fact that the SARS-CoV-2 virus, of course, particularly the earlier variants, affected the lungs quite seriously. Now, so how do I view then our understanding of long COVID versus ME/CFS now? And some of you may have seen this jigsaw picture that I've used before. When I started my research in 2012, at the beginning of this 10-year stint, I looked at all of the research that was around, and there was some very interesting, colourful research, highly focused research project, like the interesting, colourful jigsaw pieces of a jigsaw puzzle, but it wasn't possible to see what the integrated picture was. So I actually chose to work in a holistic way, not my normal biomedical way, and mount programs where I looked at multiple classes of molecules within any ME/CFS patients, so that I could try and get an integrated picture. And I have to say, 10 years on, we do have a much more integrated picture of the disturbed physiology in ME/CFS now, and I'm very, grateful that our group has been able to contribute to that international effort to get there. But with long COVID, I see that in 2022, where we were in 2012 with ME/CFS, that we're still searching for the integrated picture. But causes of long COVID have been hypothesized, and some of you may recognize some of these. For example, it's been proposed that long COVID is an autoimmune disease. Secondly, that it occurs because the virus persists, the SARS-CoV-2 virus persists, and in those people that it persists, they get long COVID. And kind of a corollary of that is maybe it's not the SARS-CoV-2 virus, but there's a latent virus that could be reactivated, like Epstein-Barr, for example, the glandular fever virus. And third proposal has been the diseases caused by chronic inflammation. Now, many of you will recognize those three things have been raised as the primary cause of ME/CFS over the years. Certainly in my 30 years of involvement of, from a family and then researcher, I've seen those. And what we can say, they are features, they occur as features of the disease, but they're not the primary cause. The one exception was this new suggestion that both people with COVID and then long COVID have microclots resistant to degradation. This was discovered by the South African scientist Rezia Praetorius. And a very attractive hypothesis with the clotting in capillaries prevented proper blood flow, and then oxygen wouldn't get to the cells. And so that's where we would have the energy deficiency and fatigue. So that was a very attractive hypothesis. And so this could have been a specific cause for long COVID, which may not have been relevant to ME/CFS. But as time's gone on, this has now been, and some researchers haven't found microclots, but also microclots have been found in other diseases. And there's been one study in August 2022 from the Praetorius lab where they've actually found at low level microclots in ME/CFS patients, but they're saying these are likely to be a cause of the disease, not a result of the disease, not the cause of the disease. So what we're actually seeing with this is that long COVID, and I think just like ME/CFS and its many symptoms may not present as one and the same disease in every individual. And I think that means the focus in terms of the health profession needs to be more on the patient and a little bit less on the disease. And I contrast that with something like type 2 diabetes or chronic illness, where, of course, there is a molecular biomarker. And so then the clinician has tools to actually treat the person. So they're really kind of focused on the disease, and the patient fits into that group. And I think long COVID and ME/CFS are much more complex, where there needs to be much more attention on the individual. So there's one question which hasn't really been looked at in any great detail internationally, and that is, why do only a minority of people get ME/CFS when they have a viral illness or some other stressor? And long COVID, why is there only a minority of people that get long COVID after getting the SARS-CoV-2 virus? Perhaps, internationally, it's still recognized that's about 1 in 4 or 1 in 5 for long COVID. And if for something like ME/CFS that arises from glandular fever, it might be 1 in 10 or 1 in 20 people will come down with ME/CFS. There are some twins studies, evidence that for genetic susceptibility, and I think everybody kind of in the field feels there's genetic susceptibility for those people that come down with it, but the number of studies is very few, but I'll talk about a new one just in a minute. So individual genetic profile then can give susceptibility, but also I think equally likely is previous medical history. And one example might be if you've had glandular fever and you're carrying the Epstein-Barr virus in latent form, that means it's silent, that it can be reactivated by a new stress event, new viral infection, and that might be the thing which gives you the susceptibility for developing the ME/CFS type syndrome. But it may also just be your life situation. I mean, we all have periods of stress in our life, physical, emotional, and psychological, and that might just give you a high degree of susceptibility. So I'm saying that it looks like it's a mixture of nature, that is to say, what you've inherited in your DNA, whether you've drawn a bit of a short straw on that, or nurture, things that the environment has impinged on you through your life. It may be a mixture of that. So I just want to talk briefly about Anna Blair's genetic susceptibility study for ME/CFS and long COVID. So she has done a quantitative survey study on 160 ME/CFS patients and 50 long COVID patients, looking for susceptibility factors. And we're very grateful that ANZMES actually helped with the recruiting of these into the study, and also their long COVID social media group. And so she probed things like, is there anything in the personal health history of the people that have come down that one can relate to the reason that might be that they actually developed the disease? And she found, on average, each patient that was recruited had two underlying health conditions. And also, they reported they took longer time than maybe their peer group or their siblings to recover from prior illnesses in childhood, and they might have had more viral illnesses than would be normal. So the conclusion was there does look to be a link to sort of underlying health conditions and indications of body physiology not necessarily responding so well to infections. What about the genetic component? So the question was, is there clear evidence from the cohorts of these running in families? And certainly the conclusion was quite clear. There was a statistically significant association between the family health histories and the development of the individuals that develop long COVID and ME/CFS. And that might mean that an aunt who had some sort of fatigue illness may not have been diagnosed, but lots of examples within the families of similar type illnesses. One thing that came out of the survey was about understanding of the condition was that patients wanted health professionals to have better knowledge and also to have more empathy. And so I guess this is always going to be an ongoing issue when there are such these are such complex diseases. In terms of symptoms, very similar symptoms between the two cohorts. One could say with long COVID, they reported more breathing problems, ME/CFS, more food intolerances. So the overall conclusion from this was that ME/CFS and long COVID are very similar diseases according to Anna's study, and there certainly are susceptibility factors. Now, I just want to talk briefly about a study which has just come out in a preprint journal a couple of weeks ago. And this is, I consider, the best study on genetic susceptibility in ME/CFS patients so far published. It's not been reviewed yet, though. So it came from a company, Precision Life in Oxford, and they used the UK Biobank to get their patient cohort samples. And they did what's called a genome-wide association screen and looked to see if there was an association with the illness. Now, what that means is that in our 3 billion bases of DNA, what makes us stand apart from our partners, our friends, our neighbors, is that there's variation in the number of bases between individuals. And of the 3 billion, there might be up to 2 million bases, a lot in number, but only 0.1% of the total. And so they're looking to see whether any of these variations associate with the illness. And so they identified what's called 199 of them, and they're called SNPs, or single nucleotide polymorphisms, just single base changes between, from the norm, really, for what most people have. And they mapped to 14 genes, and they were significantly associated with 91% of the ME/CFS cases from the cohort they examined. And that doesn't mean that all of these people had the 199 variations. There were clusters of them, so they had combinations of some of them separating into clusters. And so this is a very definitive finding that some people, unfortunately, in their genetic makeup do draw the short straw, and that gives them susceptibility to developing the illness. They also looked to see in the genes that were the SNPs related to whether you could understand why that might underpin the development of ME/CFS. And they showed some linkage, although this is very soft data in terms of linking to these things, but a very good study. Now, I want to say just a few words about our model for ongoing disease and for relapsing, which was developed both for ME/CFS and long COVID, although the data underpinning it, certainly from my group, you know, because this was written over the summer, came almost exclusively from ME/CFS. This looks like a complex diagram, but I just want to take it through with you from the top. It's not so complex. So we're saying there's an initial event, a viral infection, for example, or with ME/CFS, it might be major surgery or toxic agricultural chemical, another microorganism, or just a stress event. That leads to a peripheral immune inflammatory response. We've certainly documented that. And that's the normal response you get when you get some external thing that attacking you, some foreign organism. For most of us, that's transitory. It disappears after a few days. It returns to normal. But in some people, it becomes chronic. And what we've proposing here, there are some routes by which this chronic inflammation can get transferred to the brain and the CNS. I won't go into what these are for this talk, but there are routes, at least 3 major routes where such signals could get to the brain. In the brain, what happens, it activates the brain's own immune system, which is a separate immune system from all of the other organs in the periphery. And that leads to fluctuating neuroinflammation. And in both long COVID now, just recently, and also with ME/CFS, neuroinflammation has been documented. And so that leads then to the long COVID symptoms, but we've also looked very carefully at what molecular mechanisms might be over this era, and I'll just speak briefly about that in a minute. And so we get our neurological symptoms. The only other thing I want to just mention is, so because, I should say, because of this fluctuating inflammation, the brain thinks and CNS thinks it's under constant threat. And so it responds in a way as if there was an acute infection and keeps responding like that. And that's what we think is why the disease is ongoing. Now, I want to just talk about this light blue box here. This is the paraventricular nucleus, a cluster of neurons in the hypothalamus, which acts as the stress center. It resolves stresses. So for most of us, if we have a minor stress each day in our life, this will be resolved with some response from the paraventricular nucleus, the neurons there. It might lead in the end to a slight increased release of cortisol, for example, or corticotropic releasing hormone, things just to protect the body from the minor stress and resolve it. But we think in ME/CFS and long COVID, every minor stress is interpreted by this PVN, which is probably affected by the neuroinflammation, as though it was a major event. And so this kind of is like stoking the fire of the ongoing disease. It's like putting more coals onto the fire so that the brain is signaling back to the peripheral system that it's in danger, and so these symptoms keep going. Now, we have now got some data on comparative studies between ME/CFS patients and age-gender matched long COVID patients compared with healthy controls. Now, I just want to show you just a couple of fairly simple plots from this data to illustrate what we've been able to do. So we looked at 4,000 immune cell proteins. So that collects a huge amount of data in individual patients with ME/CFS, with long COVID, and the healthy controls. We have an ability to stratify all of the huge data which comes out of this analysis and ask by doing what's called a principal component analysis. And this is what this diagram is. And it allows individual patients to separate out if they're different from others. And I've just, for simplicity, color-coded long COVID and ME/CFS together and the healthy controls with a different color, blue. And I think you can see that the red individual patient circles separate out beautifully from the healthy controls. And that's quite a significant, to get a result like that is a major, shows there's a major difference between the immune cell proteins in the long COVID ME groups and the healthy controls. Now, if I'd actually coded these separately, they are just sort of a random mix in this group. And so what this is saying is it looks like long COVID and ME are very similar in the changes in their immune cell proteins, and they're both very different from healthy controls. We can then take data, the individual proteins, and do what's called a string analysis. And this complex diagram here is that. So here we're looking at the long COVID group versus the healthy controls, and we're looking at the proteins that are made in higher amounts. We call them upregulated. And of the 4,000, there's about 100 that are like that. And so the individual proteins are just the individual balls. And what we're looking for is the connections between them and to see if we can identify clusters. So I've put 2 circles around here for 2 small clusters, and these are proteins that are all involved in immune function and inflammation. Here, this is all of these proteins with the connections are involved in energy production. You can see we've got 2 very dense ones down here. One's just general metabolism of the immune cell, one's it's RNA biology. I won't say anything more about that. Now, had I put the same diagram up for ME/CFS versus control, it would have been almost identical diagram. And so this is telling us that from the point of view of the immune cells, these diseases, the changes in the immune system, peripheral immune system, are very closely related. So that's quite good in terms of if all the research is going on with long COVID, that should be relevant to ME. So I just want to come back to this model now and talk a little bit about what about the mechanisms here, molecular mechanism that might be leading from the fluctuating neuroinflammation to the neurological symptoms, and just focus on one area, energy production and oxidative stress. So this is a similar sort of diagram presented in a slightly different way. So here's the chronic inflammation I talked about from the model, and here's the neuroinflammation. With this scenario, what we have is the energy factory of cells is not working quite properly. This is the red sun here. This is our energy. And actually, these are mitochondria. They're called mitochondria, and they're dysfunctional. Now, what happens when this occurs is we get an unregulated reactive oxygen species. These are damaging, and they can damage our proteins, our DNA, and our lipids. And also, if the brain's immune system is activated, and here's microglia are immune cells, kinds of immune cells in the brain and the CN, if they're activated, they also produce reactive oxygen species. So it's this damaging our macromolecules, which is the cause of oxidative stress. In the last data slide, I just want to show a part of our study on post-exertional malaise to relate to the points of energy production and oxidative stress. So in this study, what we did is with some controls and ME patients, We took blood for them before they did an exercise protocol and 24 hours afterwards, and then they did a second protocol, and then we took a third blood sample. So we had 3 blood samples before, 24 hours and 48 hours, and we looked at any energy production and oxidative stress. Lynette Hodges came down from Massey, and she actually measured their cardiac physiology of the controls in patients as they were doing the exercise. And the interesting thing is that the ME patients were different from the controls, but they were also unique in their individual responses. So their abnormal responses were all specific to the individual patients. And that's why I say it's important now for us to be doing studies on individual patients, not just kind of large groups and smoothing all of that out. So let me just show you with these kind of pictures what this means. So here's a control, and we've got 3 lines here, and you can see those lines are pretty well all coincident. So what this is saying in terms of their energy production, the exercise, that's the red and green 24, 48, there's no defects showing up here in energy production. With oxidative stress, this person had a little bit of oxidative stress, and we were looking at a biomarker on proteins, but interestingly enough, it fell after the exercise. Now, this wasn't known that you could actually reverse this oxidative stress. I read in the literature, and they said it's not clear that this is reversible. We found that it actually was reversed in the controls that we did. And now I've got 3 examples of ME patients. And what we see here is the before 24 hours and 48 hours. We're starting to see a small reduction in the ability of the energy factory to make energy here as a result of the exercise. And when we looked at oxidative stress with this person, we got an increase in oxidative stress, which was statistically significant. So that was how one patient reacted. The next patient, you see here, we got a small drop-off from before to 24 hours after exercise and energy production, and then a major shift down. So their energy production was compromised 48 hours. So that's sort of like a classic post-exertional malaise response. They had higher levels of oxidative stress initially, but we didn't actually see any more with this patient. And then the next one here I show by 24 hours, this person's energy production capacity had dropped off. And if anything, it might have improved a little bit by 48 hours. But they showed increasing oxidative stress. So you can see these 3 patients had individual responses, even though it was abnormal. And I want to just finish the talk by talking about an interaction that I had with an ME CFS patient recently who wanted me to tell me of her experience and wanted me to tell people about it, to spread, because it might have been able to help other people. So this was a patient who has had ME for a while and was pretty unwell late last year. And so someone gave her a book, and the book was by a woman called Annie Hopper, and the title was Wired for Healing. And so she read this, and this woman has developed a program called Dynamic Neural Retraining System, and I'll give you a little bit of the background in a minute. And so the New Zealand patient could access this online, and so she started this program, and she said incrementally, just a little bit at a time, over the time that she contacted me, which was about 9 or 10 months after, she was in a bad state, she said there's been incremental improvement in her energy and her symptoms. And now when she compares how she's feeling to what she was feeling before, it's quite an amazing difference. So when I hear something like this, I'm naturally a bit skeptical as a scientist, but she asked me, do you know of this person. Did you know the book? And I said, no, I haven't heard of this. So I went onto the internet and I found this was a really interesting story. Annie Hopper was a person that her stress event, which caused an ME/CFS illness in 2004, was a neck injury. And then what she developed that after that was extreme chemical hypersensitivity, which got worse and worse, and then electrosensitivity, such that she had to go and live apart from her partner, not have any friends visit her, and she was living on a houseboat with no electricity and heating because of the electrosensitivity. So life was sort of at rock bottom for her. I tried everything, was left feeling defeated. I was afraid to have hope. So what she did, because she felt her brain it was a brain injury of some sort, and so she read lots and lots of literature. And although she was a counsellor before, not a scientist or a medical person, and she's quite clear about that, the book has got a lot of brain science in it. And she came to the conclusion that the limbic system in her brain was somehow the neural connections must be disconnected or misconnected. Now, the limbic system, the midbrain's limbic system, includes the hypothalamus, it includes the hippocampus, which is our information processing center, includes the amygdala, which is the emotional center. And so she set out in this really bad state, trying to see what she could do, she said, to form proper neural connections in her limbic system. And again, incrementally for her, over quite a long period of time, gradually she got improvement to the point where today she is fully cured from her symptoms. Now, was I still skeptical? Well, the amazing thing to me was that when I looked at her own diagnosis of what was going on in her brain, it's very similar, but using different language, to what I've derived from 10 years of research. So here's a patient who was able to cure herself by asking the question and trying lots of things to try and reverse what she thought was wrong. My research has got to the point where I'm actually at that asking question point. I looked around to see if there was anyone else who attests to having done this program. I might say I've now bought the book and I'm reading it at the moment. And the thing that actually encourages me is that she doesn't claim this as a cure for everybody. She claims that, she said, this has helped me, and she has developed this program and made it available because she wants people to have the opportunity like she had to get benefit. And it's an incremental thing. It's not a whiz-bang, do a thing for 10 days and you're going to be cured. So Health Rising, the ME blog, in 2020, I discovered, had an article about a nurse who probably has had ME/CFS since 2004, was not diagnosed till 2017, and she was confined to a wheelchair in 2019. And as often happens in America, she had every possible test that you could possibly have. And then in the end, she said the conclusion was it was in her head. So someone gave her a copy of this book, and she's gone through the same kind of incremental, did the program, the dynamic neural retraining program, which is what this person ad hoc developed for herself to cure herself. She went through this, and now from being 2019 in a wheelchair, she's able to run and walk long distances now. So she's had a similar sort of experience. I'm not advocating this, but like I think everybody affected with ME, we always are looking for ways of actually reversing the symptoms. And I found this, quite remarkable. And so I just offer it, to you all, just for your interest and attention if you want to actually go further. And so thanks very much for your attention. And that's my lot for today.
Great. Thank you, Warren. We do appreciate that. And I just wanted to make a note about that final wired healing, that Dr. Ellie Stein, who's the Canadian psychiatrist that Roz has had some dealings with as well, she created the course Pathways to Improvement that ANZMES reviewed. And during that course, she has also suggested that neuroplasticity is can create some changes to the limbic system, and that the DNRS program in Australia is one worth looking into for those that are interested in that. So that's just a little bit on that, Warren.
Well, thanks for that. Because, you know, I went into this totally blind, and I was a bit reluctant to talk about it too much because there may have been some negative things around it, but you've kind of given me. Yeah,
it's basically they think that the science is sound for DNRS, and that it's based on neuroplasticity. So I wanted to thank you, Warren, for this update on the current landscape of ME and long COVID, and also all the research going on. But I also wanted to take this opportunity to sincerely thank you for all the extra work that you do as a close ally of ANZMES. You step up time and time again. You're always willing to talk to media and clarify things with me and the committee, and sign letters of support for our advocacy efforts, and also, of course, talk directly to people with ME and long COVID when they reach out to you. So it does not go unrecognised, and I want you to know how valued you are by us all here at ANZMES and the wider community as well, of course. And we appreciate you and your dedicated service to people with ME, as well as your passion and your continued research efforts. So thank you.
Can I just briefly respond to that and say that I really appreciate being able to contact you at any time, and Heather before, and have, just constant, so that basically we're on the same wavelength and on the same page, and, and I hope that can continue.
Absolutely, and it will. And I really value being able to talk to you. Are there any questions for Warren, or any comments that anyone would like to make? I've just seen comments in the chat. So thank you for a great presentation. Thank you, Warren, for an excellent update. You're a brilliant, clear speaker too. Thank you and your team for all your hard work. Thank you, Dr. Tate. Great new info, really appreciated. And thank you, Warren. Always good to hear the information and hope that you bring us. Thank you, Warren. Always a pleasure to listen to you. So that's wonderful. Are there any questions for Warren before we move on? And another thank you, Dr. Tate.